I think
Welcome, welcome everybody to our monthly on Mondays for the month of May. Today, our topic is getting clinical trial approval and running a trial. We have two wonderful speakers today who have an amazing amount of experience, so we're really fortunate to have this today. First, we have Dr. Daniela Bota. She's the medical director of the UCI Alpha Stem Cell Clinic, professor of neurology, as well as the Associate Director of Clinical Operations here at the School of Medicine. She's a neuro-oncologist. She treats primary and metastatic brain and spinal cord tumors and other neurologic complications of cancer. She earned her MD from the Carol Davila University of Medicine and Pharmacy in Bucharest and her PhD in molecular biology from USC. Her clinical and laboratory program focuses on glioblastoma and other high-grade gliomas, and she currently leads cancer vaccine clinical trials that use patient-derived tumor antigens to stimulate immune recognition of brain tumors. Her combined clinical, translational, and operational experience gives her a very practical perspective on every stage of moving a trial from concept through approval to active enrollment. We also have Dr. Brian Cummings, who's Senior Associate Dean of Faculty Development here at UCI School of Medicine. He's the co-director of the Sue and Bill Gross Stem Cell Research Center. He's a professor in the Department of Anatomy and Neurobiology, and his research program focuses on how human neural stem cell transplantation promotes functional recovery after spinal cord injury and traumatic brain injury. Working closely with Dr. Eileen Anderson, his preclinical work helped establish the scientific foundation for one of the first clinical trials using human neural stem cells in spinal cord injuries. That experience building a translational program from bench observation through preclinical validation, regulatory submission, and first in humankind trials gives him a practical view of what it takes to bring novel therapy forward. He also co-hosts this series with me, and today he's one of our featured speakers sharing lessons learned from the translational side of trial development. So I'm going to hand it over to Dr. Bota and Dr. Cummings.
And thank you so much, Dr. Parag, for your kind introductions. I think Brian and I will do a little bit of what we are doing many times in the Stem Cell Research Center, which is trying to come in the middle from a clinical perspective and a laboratory perspective and try to do a lot of translation. Because in the end, clinical trials are the art of translation, moving from the amazing discoveries in the lab to the ability to go to the next stage of experimentation, which is in human beings, and trying to find out which one of those treatments can actually prove effectiveness. So the way in which we thought that we're going to do it today, and Brian, please jump in anytime that you feel like jumping in, is that I will start with just a few concepts about how clinical trials are built, starting from the type of experimentation that's needed again in the laboratory, the type of work that the FDA is expecting in order to vet the opening of the experimentation in humans, and a little bit of practicalities about who is doing what in a clinical trials and what it takes to make a clinical trial successful. So that being said, I'm going to start sharing my slides. And hopefully right now you are seeing the appropriate screen.
Yep, you got the right one.
So, as I was mentioning, what we're going to talk about today is the translational developmental pathway, some of the developmental stages, the UCI discovery platform. I'm calling it here drugs, but you could imagine that very similar and matched pathways are involved in cell and gene therapy discovery, where most of us are spending a lot of our life, as well as in device discovery. And then maybe a little bit we're going to talk about money, because clinical trials require money and they require a lot of money. And how can we get the funds to move the studies from one area to the other. So one great term that we keep talking about and you're going to hear in translation is called de-risking. Think about the fact that many times when we translate, we want to translate only the best ideas, the ones that have the highest chance to transform themselves in effective therapies. When we say de-risking, we think about de-risking in our careers. Trust me, it's no fun to conduct a phase 3 trial just to fail at the level of the phase 3, especially if it came from your idea. So you always have to reflect on how could have you made it better. But also it's a big point for industry because when a clinical trial is $100 million, $200 million, you know that a failed clinical trial has very high costs to many companies, and those companies will avoid partnerships where they are very concerned about the risk of the therapy you are developing. So why are companies still playing on this market where there is a lot of losses? Because it's a very big market. And it's very interesting because years ago, I would have told you the biggest market is oncology and neurology. But with the new discoveries in cellular therapies, actually inflammation and immunology, it's moving up very strong and very fast. And they are becoming somewhere around the second biggest market after oncology for clinical trial discovery. I think we have a lot of strengths at UCI, and we have to be able to know how to validate and leverage our strength to move through different stages of translation and hopefully get to the level of validation in large or small, but definitive clinical trials. So if you want to think about the duration of therapies, and I think you guys have heard about this so many times that every treatment, every discovery takes 10 to 15 years to be approved. And I think this many times is a very optimistic, and I'm looking forward for Brian to tell you stories about his evaluation of how much does it take from the time that an idea becomes interesting to the idea that you can go to the last level of review, which is called the New Drug Application or NDA review with the FDA. So this is kind of the classical framework. You also have to understand that the success rate, it's very small. We are saying less than 10% from lab to market, which means that less than 10% of the therapies that make it to a phase 1 become successful. It is very far, if you want to look at small molecules, it's probably more like 0.1% of the small molecules developed will ever make to an FDA approval in the market. So what do people do? We do a lot of disease biology and target discovery, but then we need to be able to screen in very large numbers of systems and cell therapies, cell assays, 96 well plates, 364 well plates, and that kind of assay development and screening, large scale development and screening, it's usually done in specialized institutes. One of them for us is the Center of Neurotherapeutics. I try to kind of add at least some of the resources at UCI. I'm sure there are many more. They're also in the UC drug consortiums. There are resources at other UCs. But then once you have that, what you want to go is in the hit to lead and lead optimization phase. By the time that you go to the preclinical evaluation in animal models, and especially to the level of metabolism and toxicity, this is the point on which you should probably already engage the IND office, because we have many times the expertise of what the FDA have asked for other laboratories. What kind of models? How many models? Do you need to go on a primate system? Do you need to go on mouse and rat? Is your best example a monkey? And then when you go on the clinical evaluation and commercialization, even more you should have the interplay between the physician scientists and the scientists. Because most of the clinical evaluation, it's not only choosing the right condition, but also choosing your right target. Are you going to treat insomnia? Are you going to treat anxiety? Are you going to treat pain? Many conditions have all of those three just as an example together. And maybe the patient doesn't care about any of that. Maybe they care about their ability to be gainfully employed. And that will be another outcome that might be what the clinical trial needs. And finally, when you try to think about commercialization and commercialization plans, you should definitely have the bill applied innovation involved, because this is where you have to understand your market, your pricing range, your share of the population, and all the other questions that we very rarely think when we try to develop animal models. So just keep those things in mind. They are very complex. When we work, by example, on certain projects, we ask to have a project manager, just trying to keep all of those aspects together and well benchmarked. But just to go back, what is a clinical trial? And we have had those discussions many times. A clinical trial is a research study on which one or more human subjects are prospectively assigned to one intervention or more interventions. So this is the important definition. And why is this important is because we do clinical trials on different things. I already mentioned drugs, devices, cellular therapies, gene therapy, but if you think we are making behavioral studies about behavioral modifications, about diets, about different type of apps, can an application that you're using regularly could modify your behavior? We, by example, were involved at UCI in showing that ways to monitor your respiratory rate if you have asthma, if you keep being monitored and having alarms, might prevent you from having an ICU or an emergency room admission. So those are things that you need to think about, that many things that we do in the clinic actually could be clinical trials, could be research projects, could be things that, one, they have a higher level of review. So we have to think, do we need an IRB approval? Don't we need an IRB approval? But also can provoke significant knowledge, even though they don't seem to be so complex as doing a phase 1 PKPD study. So what is a human subject? It's a living individual. And important is that for the FDA, still a human subject could be a biospecimen if it's obtained from somebody that, again, it's assigned to a treatment, or even a set of genomic or genomic markers right now. And as we discussed, an intervention can be of many kinds. So now that we go back, and now you can see it all spread through, as you could see, there are a lot of things that are happening and requiring resources for getting to the clinical development. And one of the things that we talk about, which happens from the disease biology to the clinical development, it's called the valley of death. And you're going to hear this term many times, because this is a lot of work that is not always funded by federal sources, that requires a lot of investment, and which, unfortunately, many times, based on the percentages, might not end up with a lead therapeutic candidate or a clinical trial. How can we do things better? In general, is through good and strong collaboration and communication between our colleagues, between both sides of the research enterprise, but also through collaboration and communications with the regulatory agencies, including the FDA. The FDA is actually your friend in many of the things that we do. Sometimes I say the FDA is the oracle, because we ask them targeted questions and they give us vague answers. But still, those questions should be answered and not supposed, because they're going to inform the requirements that will allow you to go into a clinical trial. So I think that I convinced you that there is a lot of work on the discovery and those 4 stages that we have discussed in the preclinical discovery. I'll let Brian give classical examples of that. Some of the points, some of the slides will be available, so you can go. I tried to even split everything there. I think this is a good model for our colleagues in basic science and translational science in the laboratory when they think about what do they need to do in order, by example, to do what we call in the CERM world a PDEV grant, where you propose that you're going to take all the steps to get to an FDA interaction and possible to an IND. So here is where we come along most of the time in the IND office at UCI. And what we are hoping when somebody comes to us is that they understand that you don't need only the science and the clinical trial, you need a lot of information about how did you manufacture the drug, the device, the intervention that you have, if it's something that's manufactured. And you also have to prove to the FDA that this approach of yours is safe, or it's safe enough for the disease that you are treating. And my clear example all the time is that what I am required as a glioblastoma physician scientist to prove a safety in patients that are very severely sick, it's very different than what it would be asked to be proven as safe in a study on, let's say, acne. Very different requirements because of the benefit risk analysis. So we take all of those together, and the idea is that we will be ready for the assessment, which is the readiness assessment, making sure that we have everything that is requested for the study. We have identified more sites if UCI is not enough, and that we are ready to go there and meet with the FDA first in a pre-IND event, and then when we submit an IND, if the FDA wants to meet with us after the submission within the 30 days, then they have the privilege of asking us back, giving us instructions, and putting the clock back to the 30-day deadline. So all those terms are very important, but now you have an IND. So what do you do if you have an IND? Oh, well, let's say that you have one for a phase 1, then you do a phase 2, then you have a phase 3. It is very hard for an academic physician to move farther than a phase 2, a very small phase 2, without industry or massive state or federal funding. And this is where I think it's very important to remember that we need to be involved as scientists in forming industry partnerships. And how can we work with our applied innovation to try to form those licensing opportunities, maybe form new companies so we can fundraise if we are really determined to go with those ideas of ours as far as we can go. And to do that, we need to think a lot about commercialization. And after a drug is approved, that's a step that we all wish for, then we have to think, even when we apply for approval, how we're going to post-approval market, monitor, and support. So who is the investigator? The investigator is the person that's responsible for the conduct of the clinical trial at the trial site. Many times, it's a clinician. It has to be a clinician because you have to be aware of all the aspects of the study. You have to be able to monitor and treat the adverse events. You have to be even able to talk to the FDA in a 24-hour time frame and deal with whatever is good and especially whatever is bad. So, but the investigator never practices alone, never researches alone. It's always the responsible leader of the team, and behind the investigators are multiple co-investigators, research associates, clinical research coordinators, regulatory coordinators, budgeting, marketing, and all of the other people that make a clinical trial team complete. When we talk about the sponsor, who is the sponsor? Oh, well, if this study is coming from your own work and the funding is coming through UCI from a federal agency, or it's a pilot at UCI, you are the sponsor, and the institution, UCI, is the sponsor. But for many clinical trials where we work with industry and we are subsidized to industry, usually the industry company is the sponsor. It is very important who is the sponsor because the liability of that study, the risks of those studies, are carried by the sponsor liability. So we have to be very careful that we understand who is the sponsor and how we're going to manage the sponsorship of a study. We already know about the protocol. The protocol is completed design, methodology, statistical consideration, and organization of a study. It also needs to contain the background and rationale. What is important to also know is that protocols get amended all the time. And the reason for that is, like when you do research in the lab, when we do research in the clinic, we find out that a set of labs cannot be collected every 2 hours, or that we needed one more MRI, or there's one biomarker that we found it relevant now and we want to add it to the protocol. You cannot change what you do as the patient without getting an approval. And that's called a protocol amendment. And if you hold the IND, if you are the sponsor, that amendment has to be sent to the FDA first approved by the FDA, and then you can send it to our local IRB or central IRB for approval before you can implement it in your patient population that you are enrolling in the clinical trial. I see that everybody now knows what a randomization is because we randomize our mice and rats, not only the human beings. But this is a very important concept in clinical trials because when we don't randomize, especially if you have two arms of the study, you always risk to have biases and preferential arrangements. So then we always try at the last stages, which are usually the phase 3 trials, to randomize, but sometimes we randomize even earlier in phase 2 studies. So when we think about the regulatory agencies, it's not only the FDA. You have to remember that this is also where the DHS is involved. And a lot of the things that DHS does are related to the Office of Human Research Protection and Research Integrity. So if the FDA approves our science, the DHS looks at the correctitude of how we conduct our studies and how we protect our own subjects to make sure that ethical breaches do not happen. So we talked about sponsors. Whoever is funding the study is the sponsor. So again, going back for the last slides to the UCI, what we have here is the discovery and development. And I put a special site here for the Bill Applied Innovation because we don't talk a lot of time about them. And I think it's important to note that we need to be involved with them and to be involved with them early because the grants, especially the ones that we applied for again from CERN, are requesting us more and more not only to put our science, the laboratory science that we are planning, the early clinical research that we are planning, but also to include the economic development, the marketing assessment, the partnership development in our strategical purpose and in our specific aims. So if you want at any time to be going forward with doing your own studies, we do have all of those things at UCI. I'm sure that if you contact Dr. Anderson as the Vice Chancellor for Research of our School of Medicine Research Leadership or Stem Cell Center Research Leadership, we will be able to guide you through the appropriate institute or appropriate court that will be able to guide whichever part of those activities you are trying to take. And you could think about it as a 3-service tier. This is something that I usually give my colleagues in the Stem Cell Center to put in the budgets for the grants, because you could have a 3-tier service, very similar to what IQVIA is going to offer, depending on how much you want to be hands-on in your clinical trial. And many of the clinical trials right now, especially to get funded, they need to have a biomarker development platform that's mandatory for an NDA approval. So those will be another things that you should consider. You know a lot about our sites. This is what happens if you develop your asset, your drug or device. This is how the income is going to be split and why different parts of the income go in different sides of the licensing income generated. You have a very good solid part of the income going directly to you as the inventor. And the part for which you have to realize that maybe it's not 100% are the costs that are involved in your patent cost, timing, maintenance, and strategy. And some of those patent costs are quite large. So the more advanced you are, the more you're going to need investment from either an institution or if you're in a startup, from the startup fundraising. And with that, I want to close because I want to be mindful of the time and move it to Brian.
Okay, well, we will, if there's any questions right now, we could, if there's one for Dr. Bota, we can take that right away. And then I can give you a case example of a bench to bedside from a basic scientist point of view. No questions. Okay. Well, we will jump to sharing my screen here. And I am going to give you a kind of quick story of my bench to bedside pipeline with the caveat not speaking just as a basic scientist, but someone who's actually taken a lot of advantage of what UCI has to offer. And as Associate Dean for Faculty Development, my goal here is to let people know, basically the bottom line is the earlier you're planning for maybe being involved and translating your work from bench to bedside, the earlier you're involved, the more likely you'll be successful at that. So Dr. Bota's mentioned a little bit of this, but I want to highlight a couple of things, particularly from the stem cell side of things, as I'm also co-director of the Stem Cell Center. And one is that we now have, as of 2024, a licensed and operational good manufacturing facility on campus. And if you have a viral vector or a cell therapy that you want to manufacture, you should be talking to the folks over there. We have a new director, Stephen Lin, who's come on board with prior industry experience. So we have the capability at Irvine to manufacture things that go into a clinical trial. And we also have the Institute for Clinical and Translational Science. You can contact Eric Verlaine or Andrea Meyer about what resources are available to you, from statistical consulting to quality by design workshops or community engagement studios. And I'm going to talk a little about how those helped me in my translation. And then, as you just heard from Dr. Bota, we also have an alpha clinic on campus, one of 9 CIRM-funded alpha clinics that have the regulatory background and can assist folks with designing a trial, the clinical design, and the IND or pre-IND filings that you need in order to go forward. So with that, one other thing I would point out is that the Stem Cell Center itself, funded by CIRM, has a shared resources laboratory program. This is a shared resources network across the other CIRM-funded sites, and you can get expertise and resources in stem cell-based modeling and banking, core facilities, CRISPR editing of cell lines, training in cell culture, and that will also potentially help you in your journey towards a translation. So just as a short background, I actually started on this translational journey by accident back in 2002 when we partnered with a company called Stem Cells Inc. that provided us with a human neural stem cell product to test in animals with spinal cord injury. And this work, it started in 2002, and within a couple of years, we had actually published in PNAS that the cell therapy worked in animals, that we could improve animals after a moderate or severe spinal cord injury, that the recovery was slow to develop as the cells had to mature and differentiate, and that the recovery required integration and survival. Now, knowing some of the mechanisms of action and the backstory helped us design a clinical trial. And actually, in collaboration with Stem Cells Inc., within 5 years of that publication, we were in a first-in-man clinical trial for spinal cord injury. Then the short story there, and I'd be happy to talk with folks later, is they changed their manufacturing process. And in that manufacturing change, they lost efficacy and they went bankrupt. So that's another cautionary tale about how you want to be prepared early on and planning in advance so that you don't lose your momentum and you don't lose time. And the other thing I'll point out is that this Stem Cell Inc. work was founded on their Batten's disease and Pilates-Merzbacher's disease pipeline that was already in the same therapy in a human trial. And so it really shortened their pipeline. So you go from this basic discovery to trying to have a FDA investigation new drug application and finally into a clinical trial. And as one example, when we were doing rodents, we were doing 75,000 cells per subject, maybe 10, 20 subjects in a group, and the needing of 20 million cells. If you think about scale up then to a dose of 20 million cells per subject and maybe a couple hundred people in a clinical trial, you're going to need billions of cells. So we call this issue an issue of scale up, and it's something you want to be thinking about as you're moving towards a clinical trial. So put that in perspective. If you were to think of one of these cells as a million cells being the size of a penny, and what's the difference between stacking up a million pennies versus 10 billion? So a million pennies would stretch across Aldridge Park in the center of our campus, whereas 1 billion pennies would stack up from Los Angeles to Denver, and 10 billion is from LA to New York and back 4 times. So that type of scale up is very complicated and adds a lot of complexity. As I mentioned, fortunately, we have a GMP facility now on campus, and you will want to meet with, as you're finding efficacy in animal, you're going to want to meet with people like Dr. Minella, our GMP medical director, and Dr. Lin for GMP manufacturing, and talk about how is it that you can scale up your product and develop that into actually a therapy that you'll have enough cells to do what you want to do. So switching gears a bit from my spinal cord work, that spinal cord work gave me the background I needed, though, to work on a cell therapy for traumatic brain injury and learning lessons we had learned from that. I got a very small grant from the UK-US Collaborative Development Team that translated into a CIRM award for a translational research award. And I won't go into the details, but that took us several years then of funding to then manufacture a xeno-free product and bank it for our initial animal studies. So one thing you'll want to be thinking about early on is how can you get away from animal products and undefined serum products and things that are chemically defined and xeno-free so that you will have less translational complications. So having made that product, we then moved to our animal model of traumatic brain injury. And one example I'll show you here is the novel place recognition task. You put an animal into a confined space where there are objects that they're familiar with, and then one of those objects is moved to a novel location. So they spend time looking at those objects, and then you can map that, and you can test the efficacy of your cell product to a vehicle control and demonstrate that you have efficacy or not. And so clearly you're going to need in an animal model some endpoint or some readout of efficacy, and perhaps you want to read out early on of a mechanism of action. So one thing that happens quite often in a traumatic brain injury is that there's prolonged inflammation in the brain. I think you can appreciate here that in the TBI animal that received a vehicle injection, there's a lot of astrocytes present as shown in red here. And if we give these animals the human neural stem cell therapy, that level of inflammation is dramatically reduced or significantly reduced. And that becomes then part of your filing package as well for the FDA. And so skipping over many years of work, but just to give you an idea of the timeline, the first efficacy was in 2014 to 2016. We replicated that. And based on that replication data in animals, we then got a translational award from CIRM to manufacture or do a process development manufacture of the cell therapy and to take frozen aliquots of that product and test it in a non-human primate. So I will point out now that while we did a non-human primate, this is something you're going to want to check with the FDA in an interact meeting early on is, do you actually need to do a non-human primate model? We wanted to do a non-human primate model because we were scaling up to a brain surgery in humans, and we wanted to use MRI-guided surgery techniques to get us into an animal model. This happened during COVID, so our original design was to have 12 animals per group, and we couldn't get access to that many animals, so we had to scale back. So there's always complications and reframing of your question. That's to be expected. We collaborated with Marcel Dadi at NeoNeuron and the Southwest Primate Facility in Texas and did MRI-guided transplants of 1 million and 5 million cells at 45 days post-injury. So we're scaling the dose up, the volume up, and we're increasing the time from 9 days in rodents to 45 days in non-human primates. And I'll just show you one example of efficacy there is that sleep-wake disturbances are very common in individuals with TBI. So they complain of insomnia, daytime sleepiness, and disrupted circadian rhythms. And when we give a dose of 5 million cells of our process development product that is frozen aliquots shipped to Texas, we see improvements in daytime activity and reductions in nighttime activity and improvements in sleep. And also as a kind of a surrogate marker, I mentioned that MRI work. Using MRI, we were able to then do a follow-up MRI 3 months after transplant in these animals. And we found that the cell therapy reduced the lesion volume in these animals and increased the intact white matter. Again, endpoints that will probably be useful in a clinical trial as we move forward. So, something that you'll need to be considering as you're moving forward is what are called chemistry, manufacture, and controls, or CMC. So this was something that we did, shown at the bottom of the slide here, of a quality by design studio with the GMP facility on campus, and we present them our research protocol and meet with them and say, how can we translate this into a manufacturing protocol in GMP conditions in a way that will be sustainable and scalable. And so you meet with people with experience in this, and they give you a background, they give you advice in how you can scale up your manufacturing. And in fact, they give you things that you'll put into your pre-IND document, which is, in this example, a final certificate of analysis of what's the identity, purity, and viability of your cells, how are you testing for sterility and mycoplasma or endotoxins, and what your cutoffs and your thresholds are. So these are things that your average basic scientist isn't going to know, but when you team up with partners on campus, you can get help in doing this. Another example is while we were working on this, we met with folks at ICTS, and they set up a quality by design community engagement studio where you have patients and family members, nurses and clinicians all meet, and you present to them the background of the cell therapy or whatever drug product you're going to be using. You give them the background and what your trial design is, and you allow them to ask questions, get interviewed by experts in the field. And in fact, as a researcher, you step out of that meeting and it's led by the ICTS folks, who then write up a report for you about what the community feels about your cell therapy or whatever your therapy might be, and what the roadblocks might be to translation. So getting that information early is quite helpful in your trial design as well. And with that whole package then, we put in what's called a pre-IND meeting request with the FDA. That starts a 60-day clock ticking. You file a letter with the FDA, and you are asking them for a meeting for your product. You're proposing a timeline and an agenda for that product. And then they have 21 calendar days to respond in the affirmative or to deny your request. And then if they do respond in the affirmative, they will schedule a type B meeting with you. In my case, I put in this letter in October, and they scheduled a type B meeting for December of 2025. And at that meeting, amazingly, there were 23 FDA staff, 18 folks from manufacturing and UCI and my collaborators across the country, for a total of 41 people in a meeting. And in that meeting, what you're doing is you're giving them a pre-IND document, and I don't expect you to read this document, but basically you're limited to 10 questions you can ask the FDA, questions about your donor material, cell banking, your product manufacture plans, storage and stability questions, how you will validate your final product, and then you even go over your clinical trial synopsis and ask them questions about what do they think about that trial design. And so you have a pre-IND meeting with the FDA. They give you feedback in that meeting. And then finally, 16 years later, I've taken that feedback, and I'm now waiting to get funding for the manufacture of the final cell product and the filing of the IND. So that was a very rapid summary of 16 years worth of work. And just for your downloading pleasure at the end of this session, lessons learned from these meetings are really, you want to start your bench to bedside conversations with experienced people well before you begin your first enabling publication. So you want to start having in your mind, where do I need to go? And the best way to do that is to talk to people who've already done it. And there are a lot of people here at UCI that can help you with that. You want to build a manufacturing plan and a regulatory team years earlier than you think you do, so that they can point out to you manufacturing glitches or final changes that you're going to want to address earlier rather than later. You definitely want to be thinking about scale up and where that translational scale up could hit a roadblock, that you put together a good pre-IND team, and that requires almost a year of preparation and practice before you have that pivotal meeting with the FDA. You definitely want to lock down your intellectual property, your xeno-free culture methods, or your manufacturing of your drug product methods to make sure that you have a license to operate, that you have license agreements with collaborators or people who own the IP. If you have to retrofit or go back on any of those things, it's going to cost you years in your developmental pipeline. You want to plan for probably replication studies. In our case, we had 4 publications in support of our product. And so a single paper could be enough, but probably you're going to want to have additional publications, and that will help you get the funding you need or the investment you're going to need for the manufacture scale up. And then finally, I want to point out that the non-human primate study, although we were lucky and things turned out well, it's not always required anymore. And so you want to hold perhaps an interact meeting early on. That's an initial targeted engagement for regulatory advice meeting with the FDA to actually ask them, do I need a non-human primate study or not? And then really, as you're on this journey, you want to prepare in advance, expect complications along the way, and just be courageous and don't quit. That's my parting advice to you folks. And then I think we should open up the floor for questions.
Thank you, Dr. Cummings, and thank you, Dr. Bota. Does anyone have any questions? You can just post it in the chat or even just unmute and ask the question yourself. I actually had a question, so I was going to start off the discussion is, if you have a faculty member that came to you tomorrow with a promising idea, but no trial experience, where should they start in their first week? Like, what should they do? Like, just some basics.
They should talk to people who've already done it.
Okay.
And there are a lot of people at UCI who've already done it, probably. So depending on what domain they're in, if they're talking about a pharmaceutical or a drug product versus a cell therapy product, obviously, if it's a cell therapy product, they should contact the Stem Cell Center and say, what have you folks done? Who can we talk to? And we'll be happy to hook them up with folks. But no matter what their product is, the earlier they start talking to people who have done it, talk to people at the Beal Center, like Dr. Bota mentioned, that the Beal Center can help with figuring out the market size and whether this is something that will be feasible or not. Getting that feedback early will really help you. Another example I would say is when I did the spinal cord injury work, and with Stem Cells Inc., they were a group of industry folks who had already done clinical trials. So they taught me, you want to be studying dosing and timing. And if you're going to be dosing somebody and giving them an immunosuppressant, you want to be testing that immunosuppressant in the animal model with the therapy that you're planning on. Don't wait until the end to do that. The FDA will come back and say, have you done the combinatorial approach or not? You'll say no. And they'll say, well, we'll see you in 2 years when you complete that study. You could cut 2 years off your timeline if you'd talked to folks in advance and thought about that early. Dr. Bota, what would you add to that?
So what I would add is really depends where you are on the spectrum of faculty at UCI. I would say that if you're somebody that wants to be involved in clinical trials, as a clinical PI, as somebody working with the scientists in the campus, first of all, I think it would be very important to know who our partners are in the campus. And then I'll say that getting yourself trained. We have quite a number of training programs at UCI. Many of the professional organizations have training camping booths for people that want to learn clinical trials at all levels and with the specificity of different conditions. But it's about training. Now, I got one question from somebody that asked me directly, so I'm just going to give a general answer for everybody online because I think it's relevant. What do you do when you have conflicts of interest? And my answer is you disclose, and you disclose again. And the more that we disclose, the easier it is to manage the conflicts. Majority of the conflicts are not insurmountable. And majority of the companies actually do not have a problem with us publishing our data. at least on my experience, because many of them are startups or small companies and they want to fundraise. And the best way that they can fundraise is by making their successes public. Also in the clinical arena, some of us are, in the advisory boards or speaker bureaus for some of the companies, and that has to be also disclosed and managed appropriately. So that would be my advice. Let's disclose it.
So I would add that if you don't be worried, just ask questions, because we see conflicts of interest all the time, and they get addressed. They can be managed. Most of the time, they can be managed. And definitely, as Dr. Bota said, companies generally want you to publish, and UCI definitely wants you to publish. So they're not going to let you sign a contract that hands away your publishing rights.
So there's a question in the chat where the faculty member is working on an enzyme that is very promising in mice. The company in Japan is not interested in treating patients and are not providing manufacturing information to obtain an IND to move forward. Any advice?
Ouch. So I would say if you know what the enzyme is, I would be tempted to tweak that enzyme so that it is slightly different and go it on your own if they're not willing to provide the product. Now, so what'll happen? So this is interesting. This has in fact happened to us, where we have been working on a product that with Eileen Anderson, there was a anti-inflammatory product that was involved in the complement cascade that a drug company had developed a inhibitor for, and it worked great in spinal cord injury models of mice. And we wanted to replicate that into rats and scale up. And the drug company had been bought out by another company at that time, and the new company was doing a clinical trial for some other indication. And so when we asked for more of the drug, their answer was no. We don't want a basic science lab to screw things up, basically. They didn't want any negative data possibly interfering with their IND filing. So it's possible the Japanese company is moving forward in some indication with that enzyme. It's just not something they're telling you about. And so they're not being evil. They're being cautious in protecting their own trial work by not having data come out. So if you did a study with their enzyme and got an adverse event, you would be obligated to tell them. They'd be obligated to tell regulatory officials in Japan. So it's not unusual for them to say, we don't want you doing anything. But my workaround would be, is there an alternative enzyme or a way you could change the enzyme so that you get around the intellectual property rights to that enzyme, and now you have your own intellectual property rights.
So I would add a few more things, Dr. Kimonis, which is, please do go first to Bill Applied Innovation, because the question is this enzyme preparation that the Japanese company patented? And if it's patented, what exactly is under the patent? If it's not patented, then your patent is as good as their patent, because they don't have one, which means that the next question would be, is there any synthetical chemist that would like to process the enzyme for you? Of course, it's not simple as that. As you know, there is solubility, stability, biodistribution, all of those wonderful questions. Can it be put on a nanoparticle? Can it be maybe part of an adenovirus that can be transfected and producing it in the target tissue? But there are always those questions. It starts, however, with the intellectual property, because if the enzymatic structure and even sometimes fragments of the enzymatic structure are already patented, then you don't have the right of composition. And you might have problems being able to fundraise. Even if you can do your science, it'll be very hard to process in the clinical trials, because it'll be very hard to find industry partners if you don't have the rights, you don't have the IP.
Right. They're looking for an industry partner that will take it forward, and they're looking for royalties. It's been since September 2025 that they have promised to give me the information so we can submit to the FDA, but nothing has happened. And it doesn't look like anything will happen. I think it's being cautious, but they want the patients to have the treatment because they see that they're deteriorating, but then they're not helping me provide the information to move forward with the IND submission. They're not going to put any money into it, but they say they're not going to stop it moving forward to the patients, but nothing has happened. So yeah, I'm a little stuck right now.
Well, as Daniela said, it's
Yeah.
the risk is even if they said, well, you can do whatever you want, another company isn't going to want to step in and risk manufacture because they won't have freedom to operate.
Yeah.
Now,
We're looking for a company to manufacture enzyme. It's not easy.
do they have a US patent on this enzyme? Is it a worldwide patent or is it only in Japan?
They have enzyme for another related disease. So this enzyme is important because it crosses the blood-brain barrier. It's purely a CNS disease. So this enzyme, and we've done a lot of preclinical work. It is really impressive. So I believe it's very promising, but we're in this situation right now where we can only do the preclinical work and cannot move forward at this stage.
That is very frustrating, and it's actually kind of, so I've heard this story more than once, and it's why I actually put on my lessons learned slide is to try to make sure you have the freedom to operate early, because then you can hit a roadblock.
Yeah, obviously, yeah, we, by hindsight, should have made sure our assets were protected. All the time and effort we put into this.
Well, you should ask the Office of Research to look into what licenses were agreed to and what the MOUs were when you got the enzyme in the first place, because there might be things they're obligated to do.
I'll ask again. Thank you so much. Yeah, no, this is
You're
very
welcome.
helpful. Thank you, Brian, and I'll reach out to you to meet with you because I have a couple of other products. Thanks.
Okay, sure.
Any other questions from the group? I had one last question. This is more thinking from an institutional standpoint. Institutional review turnaround at UCI can be difficult at times for many investigators. What are realistic improvements that are in motion, and what should faculty stop waiting for and work around instead?
Ouch. I would say my first advice is you can't work around it.
Okay.
There are, IRB and escrow and regulatory review processes that just have to be done. And so really the solution is start early, start earlier than you think. And if you do hit roadblocks, contact somebody else like Dr. Bota or myself that has experience with that, and maybe they can help you move forward. But I can say that the Office of Research and IRB, they're here to help, but they can't always move at the pace we want. They only meet monthly. If you miss that deadline, you've got another 30 days to wait. So plan ahead. Dr. Bota?
Dr. Bota, do you have any thoughts or
I
advice?
think I'm coming a little bit from a different direction, and I'm saying that for investigator-initiated studies, what's the most important are actually the interactions as early as possible, informal consultations with the IRB. The risk of investigator-initiated studies, it's high, as we talked about before. The sponsor is the university. So if we want to be able to open IITs fast and to be able to open them appropriately and not go through the loops and loops, the more that we can do before the official IRB submission, the better. And that will be the quality by design studios that were mentioned with ICTS. Those will be consulting with senior investigators, and in the end, consulting with the administrators, but also the physicians involved in the IRB committees. to make sure that we are moving appropriately to step. The other thing that I'll say again for IITs, I want to remind a lot of people that in addition that with the IRB, we also have radiology safety and radiation safety and IBC and sometimes escrow, and there are other committees that do exist, and they have all of them to be addressed and applied. So those steps, I have spent many years trying to find out how to make them more practical, but I think that the only way to do that is to just confront them full on and try to take them one at a time.
To just add on that question, this alphabet soup of, things that have to get approved, go through the process here at UCI. Is there somewhere that says all the different steps or timelines for someone to do this? Like if it was a sponsored clinical trial versus investigator initiated? Is there a website that we can refer faculty to?
Yes, there is the Office of Research, and at UCI as well as for IITs, there's a lot of work done at the School of Medicine Research Development Unit, so you can find them there. Also, it's important for the physicians or the scientists that are working on clinical trials to think a little bit where their clinical trial will be conducted, because there are specific resources in the Cancer Center, in the Stem Cell Center, and for the studies that don't belong in either of them, in the Center for Clinical Research. So the earlier that resources are asked and scouted, the easier it's going to be for the studies to move forward.
Perfect. Thank you for that information. Any last questions before we wrap up for this evening? Does not look like it. Well, thank you to Dr. Bota and Dr. Cummings for taking the time for this wonderful conversation. This was, I think, extremely helpful to understand how to get a clinical trial approved in the preclinical as well as the clinical stages. So thank you for that discussion. I just wanted to remind everybody our next monthly on Mondays will be on how to fill out the AP10. It will be held on June 22nd. And our speakers will be Senior Associate Deans Kyoko Yokomori and Mohammed Helmi. And so we look forward to having all of you join. And I hope you all have a wonderful evening. And thank you again to Dr. Bota and Dr. Cummings for a wonderful discussion today.
Have a good evening.
Thank you. Have a good evening.